Yep. Okay, hi everyone. Thanks so much for coming out today. I'm going to be sharing with you my project, which is titled Exploring the Effect of Genetic Calpain Disruption on Cell Migration, Invasion, Drug Sensitivity, and Metastasis in Triple-Negative Breast Cancer. So in 2023, breast cancer accounted for over a quarter of cancer cases in Canadian women and was the second leading cause of cancer-related deaths. Oh, that doesn't want to load. Okay, that's funky. Oh no. Really? Okay, there we go. So it also skipped through my animations, but anyways. So among the breast cancer, going back a step. So breast cancer is commonly classified into the different subtypes based on the expression of hormone receptors that are commonly targeted in therapies, that being the estrogen, progesterone, and HER2 receptor. And among the breast cancer subtypes, triple-negative breast cancer is of particular concern due to its poor prognosis, which is associated with a highly metastatic phenotype. And another distinction between triple-negative breast cancer and other subtypes is that it characteristically lacks the expression of these hormone receptors that are commonly targeted in therapy. Okay, these are. It's kind of hard. It's actually out of sync. Yeah, it's totally out of sync. And also, okay. I think, does that look okay? Yeah, it's all up your animation. Yeah, that's okay. So just some other distinctions of triple-negative breast cancer from other breast cancer subtypes is that patients are usually, TNBC patients are usually younger. They have a higher mean tumor size upon diagnosis. And the TNBC 5-year survival rates with metastasis is 14% in comparison to those of other non-triple-negative breast cancers, in which 5-year survival rates with metastasis are roughly 35 to 46%. And considering that metastatic disease remains incurable, early intervention and treatment is crucial for preventing progression to metastatic disease. And next, I'll be delving in a bit more into the current treatment model. Okay, that's in sync. So upon diagnosis, TNBC patients are usually treated with neoadjuvant therapy, that being a combination of chemo with other adjunctive treatments. And the objective of this neoadjuvant therapy is to prevent spread, limit tumor growth, and determine chemosensitivity. Following surgery, which can either be a complete mastectomy or a lumpectomy, patients are usually treated with adjuvant treatment, that being either radiation or chemotherapy. And the purpose of this adjuvant treatment is to kill any remaining tumor cells and prevent recurrence. Oh, there we go. So going back to neoadjuvant therapy is often comprised of widely used chemotherapeutic agents like anthracyclines and taxanes that are given in combination with other targeted therapies, such as anti-PD-L1 for patients that are PD-L1 positive, as well as PARP inhibitors for those with BRCA mutations. And despite the success of these targeted therapies, they're often limited to specific tumor types and don't address the clinical heterogeneity that's seen in TNBC. So as a significant proportion of patients aren't able to benefit from these targeted therapies, there's an urgent need to identify other molecular targets. Oh, it's out of sync again. There we go. So there's an urgent need to identify other molecular targets to develop our other targeted treatment options for other patient subtypes. So in this interest, our lab is exploring Calpain as a therapeutic target in triple-negative breast cancer. So what are Calpains? Calpains are a family of calcium-activated cysteine proteases that are seen to be aberrantly expressed in a number of diseases, including cancer, and more specifically, triple-negative breast cancer. Pictured on the right here is a Kaplan-Meier survival analysis in which Sarah Storr and colleagues found that generally TNBC patients with higher Calpain-2 expression are seen to have poorer survival rates in comparison to TNBC patients with low Calpain-2. So considering this study and many others, are therefore the most widely studied. Other Calpain isoforms have tissue-specific expression. And the structure of Calpains 1 and 2 is composed of a conserved regulatory subunit known as Capin S1. I'm just turning on my laser pointer. A conserved regulatory subunit, Capin S1, as well as an isoform-specific catalytic subunit, which is Capin 1 and 2 and Calpains 1 and 2. And it should be noted that genetic disruption of the common regulatory subunit leads to loss of stability and catalytic activity in Capin 1 and 2. Calpains are known to have over 100 substrates that are mediated in various physiological processes such as cell proliferation, apoptosis, cell migration, and invasion. And so considering the pleiotropic activity of calpain substrates, it's unsurprising that aberrant calpain activity has been seen to contribute to chemotherapy resistance, malignant cell growth, and cancer progression. So a previous lab member of ours, Stacy Grieve, was able to demonstrate calpain's pro-metastatic activity in an MDA MB231 model. And using a resectin weight model, Stacy engrafted MDA 231 cells either with control calpain or shRNA knockdown calpain S1. She then grew the tumors to roughly 28 days before waiting another 10 days before harvesting the lungs to quantify lung metastases. Upon harvesting the lungs to quantify lung mats, Stacy found that mice engrafted with shRNA knockdown calpain S1 had significantly fewer lung metastases in compared to those engrafted with control 231 cells. And this pro-metastatic phenotype for calpain in this 231 model is likely attributed to calpain's known cleavage of proteins involved in invatopodia formation, that being PTB1, SARC, and cortactin. As well, this pro-metastatic phenotype is also likely attributed to calpain's known cleavage of proteins mediated in the focal adhesion complex, which when cleaved are involved in focal adhesion turnover. As Stacy's findings contribute to a growing body of literature supporting a pro-metastatic role for calpain in triple-negative breast cancer, we're interested in studying calpain disruption in the context or in combination with existing chemotherapies. And a potential setting for this combination would be in neoadjuvant treatment. Oh no. As I mentioned earlier, neoadjuvant chemotherapy is beneficial in terms of limiting tumor growth and determining chemosensitivity. However, a number of studies have actually demonstrated that chemotherapy prior to surgery can actually accelerate disease progression by stimulating metastasis. And next, I'll be going over a study that was conducted by a previous lab member of ours, Victoria Hoskin, in which she observed this phenomena in a 231 model while studying a calpain substrate esrin. So in Victoria's model, she saw that using a similar resectin weight model to Stacy's, she actually treated the mice with 2 doses of neoadjuvant chemotherapy prior to surgery, with either that being doxorubicin, an anthracycline, or docetaxel, which is a synthetic analog of the widely used taxane drug paclitaxel. And upon harvesting the tumors and waiting 7 more days before harvesting the lungs to quantify lung metastases, she found that in harvesting the lung mats, or in quantifying the lung mets, she saw that mice treated with neoadjuvant chemotherapy had significantly more metastasis in comparison to mice that did not receive chemotherapy prior to surgery. So in this sense, or in this setting, widely used chemotherapeutic drugs like anthracyclines and taxanes can actually exhibit dichotomous pro and anti-cancer activities. And it's actually been found that calpain disruption during paclitaxel treatment can actually sensitize cells to paclitaxel cytotoxic effects. And considering the widespread use of paclitaxel, this is a relationship that I'm interested in exploring in my project. So paclitaxel is the most widely used chemotherapeutic agent used in breast cancer treatment. It belongs to a family of drugs called microtubule targeting agents that are able to bind to the beta-tubulin subunit of either microtubule polymers or to free-floating alpha-beta tubulin heterodimers to promote stability and polymerization at the positive growing ends of microtubules. And increasing polymerization and stability, this also results in a loss of dynamic instability at the positive growing ends of microtubules that's needed for spindle assembly and mitosis. So in losing this dynamic instability, this results in activation of the spindle assembly checkpoint, resulting in abnormal exit from mitosis and subsequent apoptosis. And despite the widespread use of paclitaxel, a significant proportion of TNBC patients actually lack a pathologically complete response to taxane therapy, often associated with drug efflux or expression of beta-tubulin that's resistant to taxane binding. So considering the widespread use of paclitaxel and calpain's potential involvement in this mechanism of action, this is a relationship that I'm interested in exploring in my project. And before I get into my hypothesis and aims, I firstly just want to introduce the cell model that I'll be studying calpain in, which are AC2M2s. And AC2M2s are a highly metastatic TNBC-like cell line that actually originate here at Queen's from the Elliott Lab. in which a CBAJ mouse had a spontaneous tumor growth. This tumor was then harvested and grown up ex vivo as the SB1 cell line. SB1 cells were then engrafted into the mammary fat pad of a mouse, in which the mouse then had tumor growth and lung metastases. These lung mets were then cultured and grown up ex vivo to be then serial passage twice more, and upon the third serial passage, giving rise to AC2M2s. And Danny was able to establish CAPN-S1 knockout using a CRISPR-Cas9 system. And pictured on the left here is a Western blot showing the loss of CAPN-S1 expression in the knockouts, as well as a loss of CAPN2 expression. As I mentioned earlier, that genetic disruption of the common regulatory subunit leads to loss of stability of the catalytic subunits CAPN1 and 2. And pictured on the right here is a casein zymogram, in which this is an assay that we use to measure proteolytic activity of Calpain by looking at its cleavage of its substrate casein, which is in the gel. And the negative bands here represent Calpain-1 cleavage of casein and Calpain-2 cleavage of casein. And we see that in the CAPN-S1 knockout AC2M2s, we lose cleavage activity of Calpain-1 and 2. Now, in coming to combine this knowledge, I'm hypothesizing for my project that Calpain deficient cells will be less migratory, invasive, and metastatic than wild-type cells, and that Calpain knockout will reduce neoadjuvant paclitaxel-induced metastasis. I'm hoping to investigate my hypothesis by firstly assessing the effect of genetic Calpain disruption on cell migration, invasion, and paclitaxel sensitivity in the murine TNBC cell line, AC2M2s. And secondly, I'm hoping to investigate the effect of Calpain disruption on neoadjuvant paclitaxel-induced metastasis. Within my first aim so far, using a Presto Blue assay in which we're indirectly measuring cell viability by a measure of metabolic activity, I see that CAPN-S1 knockout AC2M2s seem to be slightly more sensitive to paclitaxel cytotoxic effects. However, this doesn't seem to be a significant difference in sensitivity to taxane. Within this aim, I've also seen that in scratch wound analysis, pictured on the right here, that CAPN-S1 knockout AC2M2s are significantly less migratory than wild-type AC2M2s. And in a representative wound closure over time, we can see that CAPN-S1 knockout cells migrate to close the wound more slowly than wild-type cells. With the addition of paclitaxel, I see that there does seem to be a trend towards decreased migration rates in the wild-type and knockout AC2M2s. However, it would appear that the genotype loss of Calpain is more significant in reducing migration rates than is this slight additive effect with paclitaxel treatment. Also within this aim, I've also conducted invasion assays, which is a scratch wound with a matrigel overlay, in which I see that there does seem to be a trend towards decreased invasiveness in the CAPN-S1 knockout AC2M2s. However, a high standard deviation and low N value doesn't reveal any significant P values. Pictured on the left here is a representative wound closure over time, in which we see that the CAPN-S1 knockout AC2M2s invade into the wound more slowly than wild-type cells. With the addition of drug, I see that there does seem to be a trend towards, or a slight trend towards decreased invasiveness with the addition of paclitaxel, but again, very high standard deviations and low N values don't reveal any significant differences between the wild type and CAPN-S1 knockout AC2M2s at the various treatment concentrations. General conclusions for this aim so far would be that in my MTT assay, Calpain deficient cells are slightly more sensitive to paclitaxel treatment than wild-type AC2M2s. As well, in my scratch wound analysis, I see that Calpain deficient cells are less migratory than wild-type cells and that there may be a slight additive effect with paclitaxel treatment. Within my invasion assays, I see that Calpain deficient cells trend towards decreased invasiveness. However, more work is still needed to be done to reveal any potential statistical significance. Now moving on to my second aim, which is to investigate the effect of Calpain disruption on neoadjuvant paclitaxel-induced metastasis. So before getting to study Calpain's potential involvement in this phenomena, Danny and I firstly wanted to establish the innate metastatic phenotype of wild-type and knockout AC2M2s, and we did so using a similar resectin weight model to Stacy's experiment. So by engrafting 7,500 wild type or knockout AC2M2s into the fourth mammary fat pad of 8 to 12-week-old RAG2 mice, we then grow the tumors to roughly 400 millimeters cubed in size, and then harvest the tumors and wait another 10 days before sacrificing the mice and harvesting the lungs. Pictured on the right here are the tumor growth rates over time, in which we see generally that the wild type and knockout engrafted mice, their tumors grow at quite similar rates. Upon harvesting the tumors, we see no significant differences in terms of mean tumor sizes upon resection. However, upon harvesting the lungs, we see that mice engrafted with Calpain S1 knockout AC2M2s have significantly less metastatic burden than wild-type engrafted mice. And again, this is consistent with Stacy's findings in a 231 model in which she saw that shRNA knockdown Calpain S1 231s were significantly less metastatic than the wild-type cells. Now, having established this pro-metastatic phenotype for Calpain in our AC2M2 model, we next wanted to establish whether or not paclitaxel could elicit neoadjuvant chemotherapy-induced metastasis. And to do so, we followed a similar resectin weight model to our last experiment, but treated the mice with various either DMSO or various concentrations of paclitaxel 24 hours prior to tumor resection, before again harvesting the tumors and quantifying the lung mets. Pictured on the right here are the tumor growth rates over time, in which the red arrow indicates the date that we gave DMSO or paclitaxel. Upon harvesting the tumors, we saw no differences in terms of tumor resection sizes, which is as expected as we were only treating the mice 24 hours before resection. However, upon harvesting the lungs, we see that there was a trend towards increased percent metastatic burden with the mice treated with increasing paclitaxel concentrations, and that this seemed to be most apparent at the 9 mg per kg treatment concentration. So in having established that neoadjuvant paclitaxel could elicit chemotherapy-induced metastasis, we decided to go forward with a 9 mg per kg treatment concentration to study Calpain's potential involvement in this phenomena. In our final resectin weight model, we wanted to mimic Victoria's treatment model, in which we gave the mice 2 doses of neoadjuvant paclitaxel, the first of that being on 5 days before tumor resection, when tumors were palpable at roughly 80 millimeters cubed. And again, we gave a second dose 24 hours prior to tumor resection. We then similarly harvested the tumors and then waited only about 7 days before harvesting the lungs to quantify lung metastases. Pictured on the right is the tumor growth rates over time, and the red arrows indicate the date of DMSO or paclitaxel treatment. Upon harvesting the tumors, we saw a trend towards decreased mean tumor sizes between the DMSO and paclitaxel treated mice, and saw that this was actually a significant difference in the mice engrafted with Calpain S1 knockout AC2M2s. And again, this is consistent with our model in which Calpain deficient cells are more sensitive to paclitaxel cytotoxic effects. However, upon harvesting the lungs to quantify lung metastases, we saw no significant differences between the genotype groups and there was also no significant differences between the DMSO and paclitaxel treated mice. Conclusions for this aim so far would be that Calpain deficiency in AC2M2s impedes its metastatic phenotype, and that 1 dose of neoadjuvant paclitaxel increases metastasis in our AC2M2 model. However, 2 doses of neoadjuvant paclitaxel had no effect on metastasis. It did reduce tumor volumes in mice engrafted with Calpain S1 knockout AC2M2s. So in coming to conclude this aim and rethinking our model and understanding of Calpain and its pleiotropic protein substrates, we were wondering whether we could target a microtubule binding protein that is also a Calpain substrate. We then asked the question, can we further promote paclitaxel-induced microtubule stability in our Calpain deficient cells by targeting a microtubule binding protein that's also a Calpain substrate? And this was when I became interested in collapsing response mediator protein 2, or CRIMP2. CRIMP2 is a microtubule binding protein as well as a Calpain substrate. And CRIMP2 belongs to a family of 5 phosphoproteins, CRIMPs 1 to 5, that are known, with CRIMPs 1 to 4 being known Calpain substrates. CRIMPs are ubiquitously expressed. However, CRIMP2 is the most widely studied isoform due to its more abundant expression in the adult nervous system. CRIMP functions largely in neuron polarity, axon guidance, and regeneration, but what I'm interested in for my project is its tubulin binding and microtubule stabilization ability. CRIMP structure is composed of an N-terminus, as well as an alpha-beta barrel domain, and most importantly, a flexible and unstructured C-terminus tail. The C-terminus tail is required for microtubule binding, for microtubule polymerization, as well for binding free-floating alpha-beta tubulin dimers to promote polymerization at the positive growing ends of microtubules. The C-term also has a number of phosphorylation sites, which regulate its microtubule binding abilities. As well, the C-term also has a Calpain cleavage site at the serine 517, 518 site. Cleavage of CRIMP by Calpain disrupts its microtubule binding abilities. And I also want to note that cleavage of CRIMP by Calpain also exposes a nuclear localization signal upstream of the fair phosphorylation site at the 472, 471 amino acids. So, in general takeaways from this slide is that CRIMP is a microtubule binding protein, and Calpain cleaves CRIMP, therefore disrupting its microtubule binding ability. Just wanting to provide a bit more of a visual aid to our rationale here, we understand that paclitaxel binds to the microtubule lattice, promoting its microtubule stabilizing activity, and then it also binds to free-floating alpha-beta tubulin heterodimers to promote polymerization at the positive growing ends. Similarly, CRIMP also binds to the microtubule lattice to promote stability in its homotetrameric form, and as well, CRIMP also binds to free-floating alpha-beta tubulin heterodimers to promote polymerization at the positive growing ends. And knowing that Calpain cleaves CRIMP on its C-terminus, therefore disrupting its polymerizing activity, as well as its microtubule stabilizing activity, we're wondering whether Calpain disruption in our AC2M2 system could potentially promote CRIMP-mediated microtubule stability during paclitaxel treatment. Our hope is that this enhanced microtubule stability could further limit microtubule-mediated cytoskeletal remodeling and cell motility, as well as enhance paclitaxel's cytotoxic effects by further promoting microtubule stability. So in coming to combine this knowledge, I'm also hypothesizing in my project that CRIMP overexpression combined with Calpain knockout could actually sensitize TNBC cells to paclitaxel could sensitize TNBC cells to paclitaxel-induced cell death, as well as impede migration and invasion in vitro. And to investigate this hypothesis, I'm hoping to explore the combined effect of Calpain disruption, CRIMP overexpression, and paclitaxel on AC2M2 cell viability, migration, and invasion in vitro. In this aim so far, I've been able to generate CRIMP overexpression in my AC2M2s using a PMSCV Puro retroviral system, and this resulted in roughly a threefold increase in CRIMP expression in my wild-type AC2M2s, as well as roughly a fourfold increase in CRIMP expression in my knockout cells. Upon looking at plate confluence over time, I see that there is no differences in terms of the proliferation rates between the CRIMP overexpressing and parental AC2M2s. And that with the addition of paclitaxel, there was also no differences in terms of proliferation rates. Also within this aim, I've seen that in scratch wound analysis that in the absence of drug, that the CAPN-S1 knockout AC2M2s remain to be the least migratory in comparison to the CRIMP overexpressing cells. And when looking at their normalized wound closure constants, I see that the CRIMP overexpressing cells are slightly more migratory than the parental AC2M2s. With the addition of paclitaxel, we see that at low doses, paclitaxel treatment may actually enhance this difference in migration phenotypes between the parental and CRIMP overexpressing cells. However, a high standard deviation and low N value doesn't reveal any statistical significance. Also within this aim, using my invasion assay, which is a scratch wound with the Matrigel overlay, I see that in the absence of drug, the CRIMP overexpressing cells trend to be slightly less invasive than the parental AC2M2s. However, and that this remains to be true at very low doses of paclitaxel, slightly higher doses of paclitaxel of roughly 2 nanomolars may actually increase the invasive rates of the increase invasion rates in the crimp overexpressing AC2M2s. Another goal of mine in this aim is to actually observe CRIMP2 localization in my AC2M2s using an M-scarlet tagged CRIMP2 construct. And using fluorescent microscopy, I've seen that in my wild-type AC2M2s, CRIMP2 signal seems to be arranged in a more distinct, that's not a word, distinct and rigid localization pattern that could perhaps suggest its localization to some sort of cytoskeletal structure. However, crimp expression in my knockout AC2M2s is generally more diffuse, and we don't see these distinct localization patterns. Upon comparing these CRIMP2 localization patterns in my wild type cells compared to antitubulin staining and other triple negative breast cancer cells, I see that these localization patterns may be somewhat similar, suggesting that perhaps CRIMP2 in the wild type AC2M2s is co-localizing with the microtubule cytoskeleton. However, this is a very small sample size and only one experimental replicate, so don't want to draw any major conclusions off these preliminary findings. Conclusions for this third and final aim would be that crimp overexpression doesn't significantly impact AC2M2 invasion. However, it may actually rescue the migration phenotype that's lost with Calpain knockout. I also see that loss of Calpain is generally more impactful in terms of migration and invasion than is crimp overexpression. And lastly, in my fluorescent microscopy pictures, preliminary findings suggest that CRIMP2 may actually co-localize with the microtubule cytoskeleton in wild-type AC2M2s, but not in the knockouts. Future directions for my project and my first aim would be to optimize and reproduce cell viability, migration, and invasion assays. As well, in my second aim, Danny and I are also hoping to optimize the timing for a one-dose neoadjuvant paclitaxel treatment, as that is when we saw significant differences in terms of neoadjuvant chemotherapy-induced metastasis. And lastly, in my third aim, I'm hoping to demonstrate Calpain cleavage of CRIMP2 in my AC2M2s, as well as optimize and reproduce migration and invasion assays, assess paclitaxel's cytotoxic effects on the CRIMP overexpressing cells, and lastly, optimize fluorescent microscopy by comparing CRIMP2 signal to an M-scarlet control. So big picture summaries, whoa, that's not a word, also summaries. Big picture conclusions would be that previous literature and work from our lab has established Calpain's pro-metastatic phenotype in MDA MB231 cells. However, our findings have confirmed this pro-metastatic phenotype in the highly metastatic TNBC cell line AC2M2s. However, these findings have largely relied on genetic Calpain disruption, and so ideally, we would like to develop a pharmacological Calpain inhibitor, as this would be more accurate to what we would be utilizing in clinic if there was any clinical translation of these findings. Oh, I forgot to add that. So that leads me to the challenges of my project. So despite the fact that there's a number of active site and allosteric Calpain inhibitors, they're generally non-specific, as they're seen to also inhibit the proteolytic activity of other members of the proteasome. And so the development of a efficacious and specific pharmacological Calpain inhibitor will be significant in the potential clinical translation of these findings. The significance of my project aims to characterize Calpain's potential as a therapeutic target in TNBC. And it is our hope that in coming to build our understanding of Calpain's role in triple negative breast cancer, that we could ideally combine it with existing chemotherapeutic agents for potentially synergistic effects, hopefully for synergistic effect to ultimately improve outcomes for TNBC patients. And I just wanted to give a huge thank you to my lab members, Danny and Yvonne, and of course to Peter and my supervisory committee members, Dr. Nickel and Dr. Craig. Thank you.
I think Yvonne's gonna have some.
That it was supposed to be a transition, but
Questions to say? There's a hand starting to emerge there.
Yeah.
So you're talking about mostly Calpain two. You kind of.
Well,
Well,
sorry, go ahead.
like looking at inhibiting Calpain two, and I guess also Calpain one because you're looking at small subunit. So immediately, what I would think of when I'm thinking of inhibiting that would just be, why not use some kind of alphastatin like peptide or changing a regulation of alphastatin. I'm just curious why you haven't looked at, or why you're not looking at that protein.
So I think Yvonne could probably answer that question better, but I think that he is looking at utilizing a peptide sequence from calpastatin to develop a protac that would be, yeah. So I think that is an avenue that Yvonne's exploring, looking at using a peptide sequence from the either subdomain A or C, and I know subdomains A and C, subdomain B, right? Yeah.
She's been reading your notebook at night.
Yeah, so that is something that Yvonne's looking at. Jane,
So,
thanks.
I don't know much about Calpains, but I was wondering if you knock it out, is there any other adverse effects that you could expect to see in the patient?
Yeah, so knocking it out because it's ubiquitously expressed, I think timing and location of calpain disruption is really important. Also because it has so many substrates, systemic calpain inhibition would definitely be an issue. And then in terms of knocking it out, we know that CAPN2 and CAPN-S1 mutations that are loss of function are embryonic lethal. So it's needed for sex determination, germline differentiation, and neuronal development. So knocking it out would also definitely be an issue in terms of that.
Okay, cool, thank you.
Wati,
Great talk.
thanks.
More clarification. I got lost with the crack too.
Yeah.
So that's so Captain S1 activates Captain 1 and 2. And then those will activate cramp 2, right?
So CAPN-S1 and CAPN2 combined together make calpain-2, and then CAPN-1 plus CAPN-S1 makes calpain-1. But in disrupting CAPN-S1, we lose calpain-1 and 2 proteolytic activity, and calpains 1 and 2 are known to cleave crimp, therefore inactivating its microtubule binding activity.
Go ahead, Matthew.
Also a question about crimp. The findings that you're finding here, I guess, would they be more useful for like a biomarker setting where if a patient has high crimp2, then they may be more susceptible to like CAPN-based therapies, or would you actually try and like have a drug that enhances the activity of crimp2 or makes a patient overexpress it?
I think we're definitely far off from that, considering the findings are like not what we aren't consistent with the hypothesis, but I think that could be useful either way in terms of a biomarker, whether or not a crimp high expression of crimp in a patient, whether or not that would mean that they'd be sensitive to like taxane therapies would definitely be interesting. Yeah, wait, sorry, what was the second part of your question?
Yeah, either a biomarker or an actual drug to enhance the activity of.
Yeah, I think that there's actually there's a couple of CRIM2 agonists. I think one of them is actually a UTI medication. So the development of like an actual CRIM2 specific drug, I think that there's a couple studies looking at clinical trials using that UTI medication. Well, I know that there's drugs out there that enhance CRIM2 function, but whether or not these findings support whether that actually be useful in breast cancer is a consideration. But yeah, I don't know if that answered your question, but
Yeah.
okay.
I'm just not super familiar with the like scarlet assay thing.
Yeah.
So the green one, you wanted it like you're expecting it to look similar to like blood, so it's not the knockout, or what are you?
Well, okay, expectations would have been that, I also forgot to point out, but so as I mentioned earlier, crimp is cleaved on the C-term by Calpain, and that exposes a nuclear localization signal. So the crimp signal that we see in the nucleus is what we would have expected. I guess I wasn't expecting that there would be such defined like crimp structures in the wild types, but not in the knockouts, assuming that if it's all getting cleaved, that it would go to the nucleus. And the green is antitubulin staining of immunofluorescence. So that's looking at endogenous protein in comparison to the M scarlet construct it's essentially like overexpression of crimp, but I've tagged it with a red protein. So I guess it's not endogenous protein, but that would be endogenous tubulin.
Bonnie, what? Cell lines, sorry, are these cell lines heterogeneous or is it just like one?
So the knockouts are a clone, and then the crimp expressing cells are heterogeneous because I don't have a drug selection marker on that construct. So the expression of crimp is variable within these cells. And yeah, wanting to look at the amount of signal is definitely something that's iffy here, because when you play around with the laser intensity and brightness and contrast, you don't actually know if it's a difference in the amount of protein or whether or not it's a difference in terms of, yeah, the fluorophore citation. Something like
If you
that.
look carefully, you'll see there's some nuclei in both panels that don't have any red around them, so they're transduced for the virus, but pretty good transduction efficiencies.
I didn't check.
Yeah, great talk. I have a couple of questions if I could. So you began with talking about the biomarker utility of Calpain. So high Calpain is not good, right?
No, it's not.
So where is that Calpain measured?
Within the tumor. So I think in that study, yeah, so in this Kaplan-Meier survival analysis, they were looking at Calpain expression based on IHC staining in the tumor. And then I think high Calpain 2 and low Calpain 2 was just relative to what they saw in that cohort. I think there was like 200 patients out of 280 of which had high Calpain 2.
So is there any sense as to whether the tumorigenic nature or the things that promote metastasis are due to substrates within the cell or extracellularly or both?
I'm not sure about extracellularly, but wait, sorry, whether or not
So
the
there's these hundreds of substrates or more probably. So is there any idea where the substrate specificity might be that drives the activities that you see reflected here?
right. So Calpain has a number of substrates, but it doesn't have. Wait, sorry, I guess I'm confused a bit with the question, whether or not the substrate specificity relates to the metastatic phenotype.
Yeah.
Yes, in the sense that,
We finally stopped.
To think it over, but let's move on to another. So the knockout is combined 1 and 2 knockout, basically, this is different. So does that matter, really? I mean, do we know whether 1 and 2 have different effects that you're missing with the double knockout?
I mean, there is conflicting literature for pro and anti-tumorigenic roles for Calpain's 1 and 2, but I think I don't have the paper to show it, but I think Stacy Grieve in Yvonne's presentation, he showed that Calpain 1 and 2 knockout in grafted mice had slightly reduced metastasis, but it was the combination of CAP-NS1 knockout, so that combination of loss of Calpain 1 and 2 that was most significant in mitigating or reducing metastasis.
Another question. So this cell line AC2M2, that's a triple negative.
Yeah.
So I'm not sure you said that, maybe you did, I missed it, sorry. Could you slide number 25?
Yes.
24. Yeah. I don't think about microtubules very much, and even though a million times more about them than I do. But what happened here on the right-hand side with this diagram down to apoptosis? I didn't follow that at all. So how does that happen?
Right, so we know that paclitaxel exerts its cytotoxic effects by promoting microtubule stability and by having that sustained polymerization, sustained stabilizing effect. There's not dynamic instability, and that results in spindle assembly checkpoint activation, and that's how paclitaxel exerts its cytotoxic effects. So our rationale here was that can we further enhance that sustained microtubule stability and polymerization to further enhance microtubules, because that's how paclitaxel exerts its cytotoxic effects. So, sorry, were you referring to this portion?
Yeah, yeah. So if you arrest mitosis because you've got microtubule stability, does that result in apoptosis?
Yeah, so it was that abnormal exit from mitosis results in induction of apoptosis because it's kind of just stuck in mitosis. It's just results in, yeah.
And then last question. So you talk about migration and invasion a lot in your presentation. Are they the same thing? Or do they involve different mechanisms?
They're the same when we consider it in a cell model, but well, actually, I don't know. That's a pretty general statement. Like, they both equally contribute to metastasis. So when we look at migration and invasion in my cell culture model, I guess I'm just trying to look at both sides of metastasis so that 2D migration in a 2D plane versus invasion would be considering another.
So the experimental setups are different. I mean, if a tumor migrates, it must invade it. I don't know.
I think migration, like along a surface, but then considering invasions, so how it moves into the underlying like basement membrane or in its surroundings.
Thank you.
The invasion migration thing. Yvonne and I are both proud owners of Rumba vacuum cleaners. And that is my favorite metaphor for the difference between migration and invasion.
Great.
It migrates, but
You don't want to invade it.
it can't invade.
It's not good.
Any other questions? I don't see anything on the Zoom.
No.
If not, then join me in thanking Sam for